present my PhD thesis, which was done at the Jožef Stefan Institute in Ljubljana, Slovenia, under the supervision of Professor Igor Križaj. And as you mentioned, this will be a completely different topic and a little bit different from what your audience is used to. But I hope you will still find it interesting to see how we started by studying the beta-neurotoxin from snake venom, but we ended up knowing a little bit more about mammalian phospholipase and maybe even learning something about Alzheimer's disease. But let's start with the basics. So secreted phospholipases, which we worked with, they're one group of phospholipases A2, which are enzymes that cleave phospholipids in the position SN2. So they release free fatty acid and lysophospholipid. So this is their enzymatic action. But what they don't teach you in school is that phospholipases don't only act as enzymes, but also as ligands. They bind to a group of very diverse proteins, and many of their effects are actually a result of this binding. And if this sounds interesting, I invite you to check out our review paper, which covers exactly that topic. In our experimental work, we focused mainly on these two phospholipases, amoditoxin and mammalian group 2 phospholipase. Amoditoxin is a beta-neurotoxin from, as you said, famous nose-horned viper, Venom. And if there are any non-toxinologists listening or will be listening, beta-neurotoxin means that it acts on the neuromuscular junctions, and by damaging the neural ends, it inhibits the release of acetylcholine, which leads to paralysis of the prey or victim. And amoditoxin has homologs, or better to say, orthologues in all mammalian species. So all mammalian genomes also have gene for homologous group 2 phospholipase. And in humans, this phospholipase is mostly associated with inflammatory role, and it also has some antibacterial effect. In a healthy brain, it is associated with outgrowth of neuroids in the developing brain and also with one type of neurotransmission. Unfortunately, this enzyme is also associated with many pathologies. And most interesting for us, its activity and amount were found elevated in the Alzheimer's disease patients in the brain regions most affected by the disease. In vitro, it was shown that astrocytes and microglial cells, when activated by pro-inflammatory signals, they produce and release elevated amount of this phospholipase, which is then toxic to neurons. So interestingly, both neurotoxin from snake venom and its mammalian homologue can, in certain circumstances, have very similar effects, toxic effects on neurons. The question, however, is how this happens on the molecular level. And beta-neurotoxins are relatively well studied also on the molecular level. The process of beta-neurotoxicity is super complex, but the bigger picture is well known. And one of the main events in the process is the interaction of the toxin with the mitochondrion. Amoditoxin was shown to internalize into motor neurons, also cultured cells, to associate with mitochondria and cause their swelling and complete disruption of their structure. Mammalian group 2 phospholipase can have similar toxic effects on mitochondria, which was mostly shown for extracellular mitochondria. However, the same enzyme was also shown to reside inside the neuronal mitochondria, where it was absolutely not harmful to this organelle. And what exactly it does there, still nobody knows. But in any case, the interaction of this type of phospholipase So in any case, the interaction of this type of phospholipases with mitochondria seems very intriguing, and we focused on these two, on the amoditoxin and red group 2 phospholipase, and their effects on neuronal mitochondria, and whether that effects share the same or similar molecular background. So previously, we found that amoditoxin targets cytochrome C oxidase in neuronal mitochondria. Cytochrome C oxidase is a respiratory chain complex, also known as complex 4. And by binding to it, amoditoxin also inhibits its oxidase activity. So this suggested the probable mechanism of mitochondrial degradation in the process of beta-neurotoxicity. But maybe even more importantly, it opened a new question. What about the mammalian group 2 phospholipase? Does it, since it's a amoditoxin homologue, does it interact with the same target? And we tested this on the mitochondrial membrane fraction isolated from porcine brain cortex by doing so-called heterologous competition, where we looked into whether red group 2 phospholipase competes with amoditoxin for the binding to the same mitochondrial receptor. And we found that there is a competition, and this strongly suggests that red group 2 phospholipase binds to the same target, although with lower affinity. We also confirmed this interaction with the alternative method pull down. We combined affinity chromatography with mass spectrometry, and we found the same target, cytochrome C oxidase subunit 2. So these results strongly suggested that also the red group 2 phospholipase interacts with the same mitochondrial target as amoditoxin. And the next question, of course, was what about the effect? Is it the same as in the case of amoditoxin? And we tested this on isolated mitochondria from PC12 cells. We treated mitochondria with phospholipases and then spectrophotometrically measured oxidase activity. And we found that, a bit surprisingly, red group 2 phospholipase substantially inhibited the oxidase activity. And we also found that enzymatically inactive mutant of the phospholipase had basically the same effect. I didn't mention, but in all our experiments, we used recombinantly produced phospholipases, and we also, besides the active phospholipase, we produced the enzymatically inactive mutant, which has a mutation in the point mutation in the active site, which renders the phospholipase enzymatically inactive. So the result of the mutant, which is basically same as the active phospholipase, suggests that this effect is a consequence of the binding activity of the phospholipase and not its enzymatic activity. Next, we looked into the effects of phospholipases on mitochondrial function in cell level. More specifically, we used flow cytometry to look into their effect on mitochondrial membrane potential, and we found that both amoditoxin and red group 2 phospholipase slightly but significantly lower the mitochondrial membrane potential. And we did something similar also on the red brain tissue sections, which is a kind of a ex vivo experiment where we histochemically found again that both phospholipases slightly but significantly affect cytochrome C oxidase activity. The last part were localization studies, where we used transmission electron microscopy and confocal microscopy with corresponding labeling of the phospholipases to investigate whether the amoditoxin and red group 2 phospholipase use the same or similar pathway for internalization into the cells and translocation to mitochondria. And we found that both amoditoxin and red group 2 phospholipase do not require their enzymatic activity, their phospholipase activity to internalize into the cells and reach mitochondria. And also based on their localizations, we can conclude that they most probably do use a similar pathway for internalization and translocation towards mitochondria, and also into mitochondria. And combined with all other results, to summarize, our results suggest that amoditoxin and mammalian group 2 phospholipase affect neuronal mitochondria based on similar molecular background. And we also went a step further, and based on these results, we formed a new hypothesis on how group 2 phospholipase is involved in Alzheimer's disease. So, as I already mentioned in the introduction, we know that inflammation activates astrocytes, and they then produce elevated amounts of group 2 phospholipase, which is secreted, and then we hypothesize what happens next is very similar to what happens in the process of β-neurotoxicity during the intoxication. So the group 2 phospholipase, because of the elevated amounts in concentration, is internalized into the neurons. It reaches mitochondria, where it binds to cytochrome C oxidase and impairs its activity. And what we also know is that impaired respiratory activity leads to more ROS production, more lipid peroxidation, elevated amyloid beta and protein tau aggregation, and all of these in turn leads to even more respiratory impairment. So we have a little positive feedback loop on the cell level, and all of this also leads to more inflammation, which in turn activates even more astrocytes, then they produce more phospholipase and so on. So we also have a positive feedback loop on the tissue level, and all of this leads to more neurodegeneration. So this is only a hypothesis on its own. It sounds pretty wild, but our results and others that I mentioned suggest that at least maybe it's worth looking into, and I hope it will get some funding in the future and it will get investigated. And that's all. I would like to thank all the people involved in the work, mostly Igor, Jarny, and Adriana. And as you mentioned, I moved to another group, to completely different field. So I want to use this opportunity to say hi to my now ex-group. And that would be all. Thank you all for listening, and I'm open for any questions or comments.
Thank you very much, Adrian. Are there directly any questions about phospholipases? And I will start with one. Do you know which part of the phospholipase is responsible for the binding with the receptor? So would it be possible to generate fractionated phospholipase, or are, for example, destroyed phospholipases still active by binding? So even not enzymatic stable anymore, would fractions still be toxic?
Yeah, we didn't go into that much detail, but we do have some old results from our group, which were obtained with amoditoxin, when the identity of this mitochondrial receptor was still not known, but nevertheless, they used some chimeric phospholipases, and they found out that most probably the C terminal of the phospholipase is involved in this binding. So yeah, we know that.
So it would be maybe possible that even inactivated phospholipases are still toxic to the receptors.
You mean
like a truncated version or something like that, so that you don't have the 3D structure?
Well, yeah, I don't know. That's possible. I mean, why not? Why not? Because based on these results that I mentioned, and also we did some in silico docking, we figured out that most probably, in at least in the case of amoditoxin, what happens is a sort of a competitive inhibition, because it seems that it binds to the same binding site as cytochrome C, on the cytochrome C oxidase. Cytochrome C is the main substrate for the cytochrome C oxidase, right? So if you truncate it, and if that part binds to the same binding site, and it sterically inhibits the binding of the substrate, then yeah, it should work.